So finally, we get to talk about this virus, or at least the virus that corresponds to this virion, adenovirus, sort of everyone's favorite virus except for me, who likes these crazy ones up here, which, yes, we will have a chance to talk about at the very end of the term. But before we talk about adenovirus, which is sort of the polyoma papilloma viruses, we keep getting bigger and bigger and bigger here. Just wanted to review those really briefly. Quote-unquote tumor viruses. Actually, that's what adenovirus is as well. Very much a quote-unquote tumor virus. Not really one of the real ones. A couple things as far as polyomavirus is concerned. Probably the main thing, it's also true for the papillomaviruses, is the genome is complexed with cellular nucleosomes, and that means that it's a great model for both transcription and replication in terms of thinking about the genomes. The papillomaviruses really are tumor viruses, and actually, in this case, I was trying to think about what's the right punctuation for the tumor viruses in this case, because some of them are tumor viruses, but it's a really, really small number, actually, overall, of the papillomaviruses that are really, truly tumor viruses. And I forget whose question it was about how they get released from the cell. The whole tumor virus, actually papillomavirus release, because they're naked virions, and how do they then get released from cells? The answer is it still seems to be very much up in the air, which is kind of why I guess I have a decent excuse for not knowing. Apparently, one of the big problems with working with mostly papillomaviruses, not so much for the polyomaviruses, is that they only grow in terminally differentiated cells. Well, it's really hard to grow a terminally differentiated cell in cell culture. And so that's the reason that it's been really tough to get the nice model system where you can study this in a good way. And so the best thing that I found was that what's happening is you have skin cells that get sloughed off. And I don't know if any people seen some of the studies. You're looking at the amount of skin that's like all over the place. It's really kind of frightening, or cool in different ways, depends on your point of view. But massive amounts of skin cells from basically everyone who's in this room and who has been in this room at any point in the last certain period of time that we won't get into. But what is probably happening is that those of these sloughed off skin cells actually really are lysing and then releasing those naked virus particles in that process. But at that point, they're not killing off the cells because they've already basically been sloughed off and released anyway, and they would have been basically dead if they hadn't become infected with viruses and continuing to go through the cell cycle. So I thought that's actually kind of a cool aspect of what's going on with those guys. So that seems to be what's happening with papillomaviruses and probably for polyomaviruses as well. Again, these papillomaviruses, some of them are tumor viruses, which is why getting HPV vaccination is a really good idea, particularly for your kids. Other things, as far as the papillomaviruses are concerned, important as far as differentiation with the polyomaviruses, because those are the kinds of questions that Stedman loves to put on exams, are the orientation of the promoters. So in polyomaviruses, how are they oriented? Back to back, kind of like lambda, facing around the genome. What's the big difference in the papillomaviruses? Yes, I could have asked this as a clicker question, but I didn't. So they're overlapping with each other, facing in the same direction on the same strand. The big difference there is really with those late promoters. What are the late promoters making? The late genes. What are the late genes for? Structural proteins. Those are all made, again, this very, very late differentiated cell type. And so it's those cell type specific, terminally differentiated keratinocytes, which have the appropriate transcriptional regulators to turn on those genes. And so that's how that's being regulated. Really smart, again, to totally over-anthropomorphize the viruses, a way to have this process take place. E6 and E7 are the classic oncogenes. You overexpress these proteins, you end up going through the cell cycle much more than you should. and so that's what leads to the uncontrolled development. And we'll talk more about that as far as the comparisons to adenovirus, which also, again, it's a DNA virus. These DNA viruses, which are normally infecting cells that are not regularly going through the cell cycle. They're not regularly going through the cell cycle. How does the virus have all the proteins it needs and all the precursors it needs to make more of its genome? So making DNA is something that only happens relatively rarely in the cell cycle. So all of these DNA viruses have to have ways of getting the cell to go through the cell cycle, or they're going to be dependent on actually infecting dividing cells. And so in this case, that's true with most of these. Again, it's really going to be stimulating the cell cycle. And we'll see that's happening with adenovirus as well. So there are questions on papilloma and polyomas, and no, I'm not going to ask you a clicker question yet. It'll be a little while. Yeah, David.
Are these guys using all cellular machinery to remove the introns?
So the question here is, are these guys using cellular machinery to remove the introns? Which guys do you mean?
Either the papillomaviruses or the polyomavirus.
Right. So these are all using cellular machinery for this. So one of the very few things that the large T antigen doesn't do is stimulate splicing. And so it just seems to be regular splicing machinery. And in fact, as we'll see today, splicing was originally discovered in studying adenovirus. And all cellular, this is all cellular machinery in terms of splicing. Yeah.
That seems like I'm going to break in your house, I'm going to use your computer and your printer, but I'm also going to make you edit my paper.
Yeah, oh, exactly. So the smaller the virus, there's more and more of that. Yeah. And then, if you're we'll go way, okay, sorry, getting way off a field here, but when we get to the pox viruses, you're just going to steal my identity. Instead of breaking in and all the rest of it, everything else you've got. So that's, again, getting a little ahead of ourselves here. But it's really interesting that splicing is one of those things that is clearly a cellular and almost completely cellular. I think there's a couple of examples of viruses regulating splicing, but for the most part, it's pretty much a cellular process. And we'll talk way more about splicing today, because that's basically what adenovirus is all about. It's all about splicing. So not just because they discovered it there, but in terms of regulating production of various different genes. Okay, so adenovirus. Linear, double-stranded DNA. So instead of the small circular genomes like you have with papilloma and polyoma, these are now linear genomes. But again, double-stranded DNA, so of course have to do all these health cycle regulatory things. Larger and more complicated than the polyoma viruses. The main thing that I mean here really has to do with the structure of the virion. The virion itself is an extremely complex particle, even though it's got this very regular, classic icosahedral shape. Splicing, again, we're going to spend a long time talking about differential splicing and how that's important for the most part, making a lot of these proteins that are involved in putting together the virion. And at the end, we'll talk a little bit about the obesity virus. Any of you heard about the obesity virus before? Okay, a couple of you. Hogwash. But we'll talk about that as we move through here. So again, splicing. I could probably just highlight splicing every time here in red for the whole lecture, because that's really one of the main things that's involved in regulating the production of the proteins that you get out of the adenovirus genome. There's also alternative tailing, so a lot of that also has to do with the splicing, because of course splicing and tailing and capping, of course, are all linked together with each other. So that process is also involved in giving you lots of different proteins from a relatively small number, actually, of transcripts. Cell cycle regulation, of course, that's what you have to do if you're a DNA virus infecting most cells, because most cells are not undergoing replication. A couple of things that I mentioned a little bit before, but we'll get back to talk more about terminal proteins. And so when you have a linear genome, you clearly have a problem, because if you're using RNA to prime your replication, that RNA primer is lost. If you're circular, that's okay. You can come back and catch it from the back end. If you're linear, you have to have some way of dealing with the ends of your genomes. And so the way that adenovirus has evolved to deal with this is their terminal proteins, which should sound really familiar based on which kinds of viruses we've talked about? Particularly the picoRNA viruses, which have a terminal protein at one end. So, and there also are these ITRs, also known as inverted terminal repeat structures, and that's all just about the DNA. That means at either end, you've got these sequences on one strand is identical from 5' to 3' as it is at the very other end of the genome, 5' to 3' going the other way. And we'll take a look at that. It turns out those are really important for replication processes. So our classic outline, okay, where they come from, what's the structure, binding and entry, but transcription and replication are where we're mostly going to spend a great deal of our time here. Release here does actually seem to be a cell lysis process. And so these are non-enveloped viruses. They are being produced by actual lysis of the cells, which they're infecting. I love this micrograph because not only does it have the adenovirus here, but also our little friend, adeno-associated virus. This is one of the small single-stranded DNA viruses that George Kaysen is going to talk about on Wednesday because I won't be here. So it's a nice introduction to that. I will ask him to post his lecture online, but I don't think he's going to be recording it. So bring your recorders for next time. And please then send me a copy of that, and I will post them on D2L or try and upload them to YouTube, as the case may be. So just a quick shout out as far as that's concerned. So where do adenoviruses come from? Everyone thought they had to do with the common cold. That is not the case. What causes most common colds? Rhinoviruses. However, originally found in adenoid tissue, and that's why they're called adenoviruses. In terms of disease, adenoviruses are really not a big deal, with one exception, and that has to do with basic training for militaries, and we'll get to that right at the end, assuming we actually get there. But much more important in terms of tools. So again, a lot like the polyomaviruses and SV40 in terms of thinking about how the cell does things and just using the virus as a tool to understand that. Again, my bright red word that should be here everywhere, splicing, originally discovered in adenovirus, studying adenovirus and particularly expression of the late proteins in adenovirus. And those late proteins are made by what's called the major late promoter, which we'll again get back to a little bit later on. Also is where people discovered how RNA polymerase II was interacting with DNA, how the Tata box was interacting, with TBP. A lot of the original studies on transcription were done with the adenovirus major late promoter. So very early, a lot of the early transcription data is really literally on this. More and more now people are using adenovirus for gene therapy purposes. So there's some really interesting studies going on. We'll talk again a little bit more about that a little bit later on. There are lots and lots of adenoviruses out there. 50 serotypes, 6 subgroups. This was a couple of years ago when I did this. There are probably way more than that right now. Many, many, many different kinds of adenoviruses out there. Again, most of them causing little to no disease as far as we can tell. Yes, they're tumor viruses, but again, this should be the quote-unquote tumor viruses, a lot like the polyomaviruses. And yes, there are a couple of chapters in here about adenoviruses. But basically, huge amounts of virus particles, really turning on all of the most important genes for regulation of the cell cycle, which we'll get back to again a little bit later on. And just as happening with pretty much all of the other DNA tumor viruses we've talked about so far, they're non-permissive cells. And so these are cells that are not making virus, because making virus is a fine, good way to go, but you're not going to be forming a cancer cell in that process. You'll be lysing or being sloughed off of the skin, etc. But in a non-permissive cell, i.e. you can't be making more of the virus, then if you've misregulated the cell cycle, then that cell can end up replicating in uncontrolled fashion, which is cancer, so forming tumors. Let's look a little bit at the structure. The structure here, in my incredibly biased opinion, is incredibly gorgeous. Lots and lots and lots of proteins that come together to make these icosahedral particles, and again, sort of a stereotypical kind of virus. In the virion, there are 11 proteins. It's a pseudo T equals 25 structure, and what does that mean when I say pseudo? Different proteins, exactly, that are involved. We have hexon proteins, and those hexon proteins are shown down here. A little harder to see here, but these trimeric proteins right here in all of the hexagons that fit together between the pentagons, which are at the 5-fold axis of symmetry. Here it's really easy to see where those are because they've got these projections coming out at the 5-fold axes of symmetry. How many 5-fold axes do you have in a regular icosahedron? 12. Here, and those pentons, as they call them, are actually made up of 3 different proteins. There's a base, fiber, and a knob. So the base is what's at the bottom. The fiber is this extension that comes out, and there's a little knob that sits at the end. And so multiple different proteins that are involved in this. Now, in fact, the last version of the Flint textbook was done, this was the figure that they had there, and we'll see the cartoon in our textbook, but basically very soon after our textbook was published, there was this 3-dimensional structure, in fact, 2 different 3-dimensional structures that were published basically back-to-back in Science magazine in 2010. So one of them is an X-ray crystal structure, the other one is a cryo-EM structure. And one of the things that was really amazing is that these things are almost superimposable on each other, and that tells us how far cryo-electron microscopy has come, literally in the last couple of years. And so here, these are each of those individual proteins. Here's the penton base, fiber, which you can't see there as well, and the knob up at the top. And then each of these prettily colored, so blue, orange, green, yellow, these are all the hexons which are between those. And you can count to get your 25 here just by going from this 5-fold axis to that 5-fold axis. 1, 2, 3, 4, 5. Don't have to change direction. So H is 5, K is 0. So T equals 25. Clearly different proteins. A pseudo T equals 25 structure. This is the other one. And in fact, curiously enough, it was the cryo-EM structure that was actually a higher resolution structure than the X-ray crystallographic structure. It's one of the very few cases that that's true. But that's partly because to get a crystal structure, you've got to crystallize the particle. Well, crystallizing something that's this big, I didn't mention it, but these are pretty darn huge virions. Again, pseudo T equals 25, pretty large. So getting that all to crystallize is really problematic. And one of the things, I'll take a look at the video in just a second here, but here is the electron density. So the actual data that you get from the electron microscope is the wire diagram here, the gray on the outside. And then the actual amino acids are what were fit into that with the model. And the great thing, of course, we know the genetic code. We know what the sequence is of the genome of the virus. We know what the sequences of all the proteins are. So we can just take each of those amino acids. We know the structure of the amino acids and just literally fit them into the structure. And if you have an amino acid, this is the model here with the blue and red, and then the actual data in gray, and these fit really incredibly well relative to each other. Here's a blow-up of that penton structure. Here's the penton base, penton fiber going up here, and then leading to the knob up here at the top. We'll take a quick look now at some of the... Sure, yeah.
Can you go back a couple slides, that little zoom in on protein? Is that one of the projection proteins?
Okay, we'll take a look at that actually in one of the movies, because there's one of the movies here actually has that. So if we... I think we need to get out of here quickly. I believe the movies were in here. There we go. So that's the one that we'll look at second. But then there's this one, which is the whole virion. And one of the things to look at here, here's our 5-fold axis of symmetry up here at the top. Then we've got each of our hexons here in between. But all of these different colors, the blue and the purple, these are all of the extra proteins which are sort of holding everything together here. And so let's take a look at the video here. Again, I think this is absolutely gorgeous, but also kind of biased. But we've got, again, each of these five-fold axes of symmetry here. And again, this is all from a cryo-EM image. Hopefully you can nicely see the five-fold axis of symmetry right there. And then each of these hexons right there, you can see nicely the separation of each of the trimers here. These have those double beta barrel structures, which it turns out you find in archaeoviruses, STIV, and also in some of the bacterial viruses like PRD1. So incredibly conserved structure, not at all conserved at the level of sequences. So I think really good evidence for how some of these viruses are related to each other over much longer periods of time. And Trevor, this is getting back to your question, hopefully. If not, we can go back and take a look at the other one. But here, what we're looking at is the, again, cryo-EM structure, which is all of the gray. And so what you're literally looking at there is the electron density after averaging all of these things together and then looking at them. And then inside that, which is basically invisible, which is kind of the point, is the modeled backbone given the structure of the protein that we know. And so you make a model, literally, by looking at the individual amino acids and then line them all up with the structure. And basically what they're trying to show here is that these things match just insanely well with each other. This is, in fact, the hexon protein. They're just spinning around here. Here you can see really nicely a couple of beta strands, all those guys sort of lined up right next to each other. And so that way you can get a really good feeling about how accurate this structure is. Here you can see a nice alpha helix. You can see all the things turning around there. So a really high resolution model they've gotten from the electron microscopy. And they spend a long time looking at that in this particular case, just again trying to convince us, although I'm rather convinced, that this is in fact a good process for looking at these viruses. I'm not the only one who thinks that adenoviruses have these really cool structures. This is one of the, I guess, finials, anyone in architecture? Whatever it is at the top of one of the buildings at the Cold Spring Harbor lab. Here's adenovirus. In fact, adenovirus was studied there. Splicing was discovered there. The guy who was working on splicing got his Nobel Prize because of working on this. One of the things that I also really appreciate is that this was when they were redoing their wastewater facility. So this is actually a gazebo on top of their whole wastewater treatment plant. Has adenovirus on the top. I don't think they're using adenovirus specifically for this. Cold Spring Harbor Labs is also where they had all of the phage courses, so working on bacteriophage, where Hershey and Chase did their work. I should say Chase and Hershey did their work there, showing that DNA was the genetic material after everyone else had shown it, but everyone then believed them once they'd done that. And I may or may not have mentioned before, they also have a clock tower on campus. And many clock towers will say north, south, east, and west around them. Any ideas what the clock tower there says? C-A-T-N-G around the outside. So great place if you're a molecular biology geek. But really fascinating research here on Long Island, about an hour away from New York City. So this is the much more boring picture that you have in your textbook of the adenovirus virion structure. I do not expect you to remember all of these names. This is absolutely ridiculous. They've just got Roman numbers just to make things even more confusing for everyone. Here's that penton base, penton fiber, and the knob at the end. Here's that hexon protein. But importantly, all these extra proteins. Remember sort of the blues and the purples in those 3D reconstructions? Those are all these extra ones that are sort of helping to fit together this whole structure. Importantly, here is, well, it circles it, so it's a little bit more obvious. TP, which is that terminal protein. Remember, it sticks to the end of the genome, here at one end of the genome, and here at the other end of the genome. You'll also note that it's not packaged nicely into nucleosomes like you have for the polyoma and papilloma viruses. This is a virus protein, which is binding to the DNA and packaging it. So larger genome, larger virus, more proteins, more of the viral proteins doing things that some of the smaller viruses have been depending on cellular proteins to do. The genome is, how many people remember how big the genomes are of polyoma and papilloma viruses? I always have to go back and look it up. They're about 5,000 to 6,000. Here are about 5 times bigger than that, 5 to 6 times bigger than that. And again, with these hundreds of different adenovirus genomes, they're all different sizes. But they all do have some similarities, and these are some of those. That they have a terminal protein covalently linked to the 5' end of the genome, which should almost immediately tell you something that's going on there. And also at the 5' end of the genome here. And then these inverted terminal repeat structures, which means that on one strand, you've got 5' to 3', one sequence. On the opposite strand at the other end, you should have the identical sequence. And that's when I say should have. You notice the little addition that I put in here, because this should be CTACA TCA, because here it's complementary to the CAT CAT. So this T actually should be slightly shifted here. That was obviously a PowerPoint to keystone issue. But yes, the sequences should be the same on the 2 strands here. They certainly are on the opposite strands. So here, if we're looking from 5' to 3', TGA, TGA, TG, TGA, TGA, TG. So these inverted repeat structures, about 100 nucleotides long at either end of the genome. Now, because you have these inverted repeat structures, what of course this means is that this end, where's my pointer? This end here can also bind over at this end. And this will turn out to be absolutely critical in terms of your genome replication a little bit later on. This is what the genome looks like. How does it get inside the cell? We have a chance. We may take a look at the BBC documentary looking at the virus infections. Anyone seen that? Yeah, very, I don't know, anthropomorphizing, I think, is one way of putting it. But I think it's a really cool animations. But again, I'm kind of biased here. So binding and entry, we're now looking here at adenovirus. We have the, not surprisingly, it's the knob at the end of the spikes at the 5-fold axis of symmetry, binds to the adenovirus and Coxsackie virus receptor, because they discovered these before they actually knew what their real functions were. Once you have this binding, then you have receptor-mediated endocytosis, gets into the endosome, and in fact can escape the endosome, and that's where you end up with the virion lacking the projections at the 5-fold axis. This guy gets transported to the nuclear membrane and only gets broken down at the nuclear membrane. Both of these processes, losing these spikes and breaking down at the nuclear membrane, are both viral proteases, which are involved in this. So unlike most of the cases so far where we've talked about viral proteases being involved in chopping up the genome in order to make things active, or chopping up the virus polyprotein in order to allow it to be functional or allow it to be released from the cell. Now we've got viral proteases which are involved in the entry process, getting the whole genome to the right place. Here's what cartoon of that looks like. Here we have interactions with the knob, here with the Coxsackie virus and adenovirus receptor. Then we have secondary interactions, and we'll talk more about this when we talk about secondary receptors. HIV is a classic case for this, where you also have interactions with the penton base. So it's not just the knob, it's the penton base as well. And it's only if you have both of these interactions that you have the adenovirus brought inside the cell. Turns out that these particular Coxsackie adenovirus receptor and these integrins, which are the secondary ones, these sort of black commas here, are common on lots and lots of different kinds of cells. And that's one of the reasons that people are so excited about using adenovirus as gene therapy agents, because they can actually get their genomes into lots of different cells. So once they get their genome in to the nucleus, then you have transcription that takes place. Transcription takes place all over the ... genome. Really kind of messy, at least it looks like here. We'll step through it piece by piece in the next couple of slides. There are 8 promoters, 12 polyA sites, and then a couple of just RNAs, which are some of the very first small RNAs, in fact, that people started to study looking at here. But we'll start out with the early promoters. Those are these here, E1A, E1B, E2, E3, and E4. So these are our early promoters. One of the things you'll notice is that all of the transcripts that are made from these are a whole bunch of different pieces. And that's all due to differential splicing that's taking place there. There are a couple of intermediate promoters. We're going to ignore them, other than just to know that they're there. And then the main promoter that people have been looking at for years and years and years is the late promoter, and there's one late promoter. There are multiple early and middle promoters. There is one and only one late promoter, also known as the major late promoter. That is put together from this tripartite leader sequences, and then all of these other sets of final messenger RNAs that are going to make all kinds of different proteins. And again, not surprisingly, these are all of your late genes, which are going to be what? Structural proteins. These are all of your structural proteins. And you'll also notice that there are a heck of a lot more in here than we have of just the pieces that are there, including things like the protease, which is what you need for entry purposes. Remember, the only time you're going to get transcription leading to translation is once you actually have that whole genome in the nucleus. So if you have to have a protease, a viral protease, in order to break down the virion before it gets released, that it better be in your capsid. It better be packaged. It better be one of those structural proteins.
Wait, so you're saying it's packaged in there already?
Yes, so the protease is packaged with all of those other things. And if you want to go back, we can go back and find the 23K protein in that cartoon. But important is that those proteases have to be packaged, because if they're not packaged, you're never going to make them.
Do they have like a similar protective? You know, there were some other elements and other viruses where there's like a pre kind of molecule that keeps it from being active at the wrong time. Do these have that too, or no? They can just basically be present within the virion without causing any trouble?
So the question here basically is, do you make a pre-protein a lot like the fusion proteins, the kinds of things we talked about for all of the enveloped viruses and fusions and things? So activity here seems to be regulated very similarly to what you see with, just take orthomyxoviruses. It's a pH-dependent process. And so if you're at low pH, you're in the endosome, that leads to proteolysis. The actual regulation of proteolysis at the nuclear membrane, that I'm less sure about. I'm not quite sure how, but it doesn't seem to be this kind of precursor protein then cleaved during a maturation process. These actually seem to be functional if you give them the right conditions. And again, the right conditions there are low pH. Yeah. Okay. So, and again, there are these little RNAs at the end here. So one of the things, again, maybe you remember, again, this is the tripartite leader sequence here, 1, 2, and 3. All of these late messenger RNAs have this tripartite leader sequence down at one end. And that's how people ended up discovering splicing in the first place. And this is the colorized image from that paper. This is Phil Sharp's paper. Phil Sharp was at MIT. He was competing with Rich Roberts. Problem with this darn phone. My fingers are too big. So this is from Phil Sharp's lab. Rich Roberts was the one who was at Cold Spring Harbor, where we had all those, clock towers of C's, ATs, and G's, and adenoviruses all over the place. Basically, they discovered almost exactly the same thing at the same time, and that was if you looked at the RNAs which were being produced, and in this case, they were looking for the hexon RNA, hexon messenger RNA. Why hexon messenger RNA? Because that's the structural protein that you have by far and away the most of. So that's where you're going to have the most RNA. And then what they did is they took that hexon messenger RNA and hybridized it to the DNA of the viral genome. So you get a whole bunch of viral DNA. That was easy because viruses replicate nicely, and adenoviruses actually replicate really well in cell culture, so you can get a ton of them. And you can get the messenger RNA for the hexon protein, because that's the one that's being made huge amounts of, and then hybridize them to each other. And in that hybridization process, what you'll see is double strands where you have the RNA binding to the DNA, and then some single-stranded pieces of DNA, but most importantly, double-stranded pieces of DNA where you don't have hybridization taking place. So hybridization, messenger RNA after it's been spliced, and then the DNA, which would be before you have splicing taking place. And so what happens there is because of this tripartite leader sequence here, 1, 2, and 3, transcript starts right over here. You make 1, you make 2, you make 3, and then splicing happens and you end up with a hexon messenger RNA. That process gives you loops. And so there's this A loop, which is what's between part 1 and part 2 of your leader, the B loop between 2 and 3, and the C loop, which is the really big one, getting you out to the hexon messenger RNA. So we'll, again, back up once again here and go through. I need to redo this slide. Hexon RNA is right here. So there's the hexon relative to the tripartite cedar. And this is to scale. Here's that extra splice. This is the loop is going to be the DNA of the genome between here and here. That's what that loop is going to be coming from. And how do you know that that's where all of these things are? Well, lots of different ways, but the important thing here really had to do with restriction endonucleases, cutting the adenovirus genome into very specific size pieces. And Rich Roberts and his lab were one of the very first labs to work on restriction endonucleases and had discovered, purified, and actually ended up selling a whole bunch of these restriction endonucleases. And Rich Roberts is one of the founders of New England Biolabs, which is where we buy a whole bunch of our restriction endonucleases from these days. So that was having all these restriction endonucleases was made it possible to get these pieces of DNA of really defined length and know where they were in the rest of the genome. And so it was multiple different ones, and it turns out that Phil Sharp, who discovered a different technique, Phil Sharp discovered the technique of what he called poly-T chromatography. So he made a chromatography column that had a whole stretch of T's on it. What is that good for? Binds to poly-A tails. And so it's a really good way of separating messenger RNAs. And so he did that, and this particular piece right here, this piece of the genome from that end to this end, turns out to be an EcoR1 fragment, the largest EcoR1 fragment that you have in the adenovirus genome. Yeah, Dave, you had a question.
What does this phrase tripartite leader mean?
So tripartite leader, all that it means is there are 3 different coding sequences that you have in the genome that are separate relative to each other, but they get spliced together. So 3 pieces get put together, and those then serve as the 5' end of all of the late messenger RNAs. And it's all because of different splicing that happens.
So the leader part is like the pre-slicing.
That's right. Now, the leader part here is upstream of where you have your translational start site. So these are all pieces that get put together, and not surprisingly, they're really good translation starts. And that's why they're being used for all of these late messenger RNAs. They also have signals for getting transport outside of the nucleus in order to get them well translated. So 2 things, transport and second, good for translation. Yeah?
So all these genes have the tripartite leader sequence.
All of the late genes have the tripartite leader sequence.
The polyomaviruses with the T antigen follow the same N-terminus.
To some extent. So again, sorry, to repeat the question. So with all of these having the same leader sequence, is that analogous to all the different T antigens having the same N-terminal sequence? So what's happening here is the splicing is, in the case of adenovirus, splicing is happening, at least for the leader sequence, in untranslated regions. Whereas splicing in the T antigens is happening all in the translated regions. And so here you're not going to always have the same N-terminus for all of the late genes because this is an untranslated region. Whereas for the T antigen, all the splicing is taking place in the coding sequences. And so then you'll always have the same N-terminal sequence. So 5' end, same for each of these transcripts. And that's going to be true for both the polyomas and the adenoviruses. But the protein sequences are going to be different in adenos, but the same in polyomas. Does that make sense? Okay. Good. So I need a breather. So that means you guys get to do clicker questions. So kind of me, isn't it? Primer for genome replication in adenovirus is most similar to the primer used for replication in invisibility. Yes. It will show up here. And if the worst comes to the worst, I actually have an alternative way of dealing with this. Hold it right there. PhiX174, SARS, polio, Ebola, bacteriophage C7. It's Monday morning. Main reason for clicking questions, give me a break. You're deprived. You can tell. Ten, 5, 39 of you. What do we think? The thoughts are, if I can get this over here. Oh, wait, hope that it's right. Do remember last time, yes, where I did a bad job of explaining it. Primer for PhiX174 is cellular primase, DNA primase.
No.
Not what happens here. SARS is what? These are coronaviruses. Remember coronaviruses? How do they start their replication? They've got a viral RNA-dependent RNA polymerase, which because of the N protein will sit down and bind to the ends. Ebola virus, same way. Bacteriophage T7, it's got its own primase, it's got everything it actually needs. RNA-dependent RNA polymerase. Did I say anything about RNA-dependent RNA polymerases in adenoviruses? No. So, what do we end up with? Polio. Why? VPG, which is a terminal protein, which is also serving as a primer. Okay, so let's switch gears and talk a little bit about the proteins which are being made. Go way back to the early proteins. What are early proteins involved in? The replication of the genome, and if you're a DNA virus particularly, regulating the cell cycle, because it's got to be involved in cell cycle processes. So here we have interactions of the first one, E1A. Not surprisingly, it's a lot like our friend from polyomaviruses. What's the answer for polyomaviruses by definition? Large T antigen, exactly. So large T antigen, E1A serves a very similar function, and most importantly here, interacting with our friend RB. So E1A interacts with RB. RB is blocking the cell cycle. E1A is interacting with RB in order to stimulate progression to the cell cycle. E1A also interacts with this same protein P300 that we talked about before. We talked about large T antigen. What does it do? It's a histone deacetylase. What does histone deacetylation do for you? Opens up chromatin, allows transcription to take place. Anybody remember what TDP is? Thank you. At least something came through from molecular biology. So interaction with title binding protein, what's that going to be important for? Stimulating transcription, and particularly stimulating transcription from the late promoters. ATF is another transcriptional activator, which is important in this whole process. But most important as far as regulation of the cell cycle is interaction with RB. What RB normally does in the cell cycle is binding to and blocking the E2 factors. E2 factors were originally discovered because they're important for regulating the E2 transcript in adenoviruses. So that's where the whole E2 factor comes from. Has nothing to do with the cell cycle, but at least when people discovered them, and that's why the name is historical, coming from, again, activation of the early 2 transcription unit in adenovirus. These also interact with histone deacetylases. So histone acetyltransferases put acetyl groups on. Histone deacetylases take them off, and so they'll compact chromatin. In the normal cell cycle, you have activation of cyclin-dependent kinases. That will phosphorylate RB. Phosphorylation of RB releases the E2 factors, which will turn on the E2 transcript if you happen to be an adenovirus infected cell, which hopefully most of your cells aren't. But in the other ones, we'll be activating S phase and leading to the rest of the cell cycle. What E1A does, just like large T antigen and many of the other early proteins we've talked about now for these small DNA viruses, interacts with RB and basically does exactly the same thing as phosphorylation would do in the cell cycle. So you've stimulated the cell cycle in this process. What's our other favorite tumor suppressor protein? P53. So, not surprisingly, E1A is also interacting with P53. What P53 normally does, so let's just ignore E1A for the time being. P53 is a transcriptional activator. It will activate MDM2, binds to P53, leads to P53 degradation. This is a way of controlling the level of P53 inside the cell, so you don't get too much P53. You get too much P53, you end up with apoptosis. So here, E1A binds to P53, blocks the activation of MDM2, and turns on the transcription of this cellular protein. And by the way, that's just ignore that. Cellular protein, which binds to MDM2 and blocks this MDM2 from interacting with P53 and getting it degraded. So the presence of E1A basically gives you lots of P53. Now, lots of P53 is okay in terms of stimulating the cell cycle, but lots of P53 is a problem if it stimulates apoptosis. So you're stimulating the cell cycle through RB and interactions with P53, but in order to block apoptosis, you need another viral protein. And that's where E1B comes in, which suppresses apoptosis. And basically that's all that it really does, is blocking this apoptotic process and then helps get rid of P53 a little bit later on. But the main things that are happening here have to do with the suppression of apoptosis. So E1A turns on P53, E1B turns it off again. So stimulation of cell cycle, but blocking of apoptosis. And so you have to have both of these genes functional in order to get normal replication, because if you don't block apoptosis, then the cell is going to basically commit suicide, and all of the viruses which are in there are going to die with it, if they're alive, and blah, 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 all kinds of other issues. So that's E1A and E1B. We'll talk about some of the other proteins a little bit later on. Replication. How do you get replication taking place? You've got your terminal protein. Your terminal protein has not a tyrosine on it, but a serine. Serines also have OHs, and those OHs basically act as your 3' OH for extension to the rest of the genome. There's a viral single-stranded DNA binding protein, which will bind to the strand which is being displaced, again, by the viral DNA polymerase. So you see, viral, all of these viral processes here, which then will extend your pre-terminal protein. The new DNA strand here is the orange strand. Your strand which is being displaced is now a single-stranded piece, again, bound to by this single-stranded DNA binding protein. Once this guy gets to the end, it can start again and go back the other way. But most importantly, this single-stranded DNA, which has just been blocked off here, you remember we have these inverted terminal repeats. So that means that this sequence here is the same as this sequence over here. And what that means is that this end of your single-stranded DNA can bind to this end of your single-stranded DNA. So you end up with this frying pan structure, which then the DNA polymerase can bind to and extend its way around that as well. So you've got two kinds of templates that the DNA polymerase can bind to, either this double-stranded linear one or single-stranded where the two ends have come back together with each other. And so these are the two processes which the virus uses in order to replicate its genome. And again, all through priming through the terminal protein. Making all of your late genes, we already mentioned here that we've got the tripartite leader sequence. This is your messenger RNA. This is the primary transcript which is made. Again, it's got these three leader sequences here, a 5' cap. The 5' cap, by the way, is made all by cellular processes. This is all happening with RNA polymerase II, the cellular splicing machinery. This cap structure, once you have 1, 2, and 3 spliced together, that gives you transport and translation. And then splicing can happen to one of these 5 different places. And then in each of those 5 different places, you also have multiple different poly-A cleavage sites, or potential poly-A cleavage sites. So you have 5 sets of genes in your late genes, which are dependent on polyadenylation happening at each of these places. And then in each of these groups, you have different 3' splice sites where you can end up with multiple different RNAs. So, and remember, when you have RNA processing, it's all happening co-transcriptionally. So you start with the RNA polymerase. That RNA polymerase is cruising along the template, puts the cap on, starts splicing, gets to one of these polyadenylation sites. In some cases, it'll cut polyadenylate there. That's going to stop transcription at that point. If it doesn't stop transcription there, then it will get to the next one, and so on and so forth. And all of these cases, if you skip this polyadenylation site, now you're going to start splicing into here unless you skip this polyadenylation site, and so on and so forth. So all the splicing is going to happen into that last set of potential messenger RNAs. All of these guys have different sequences, different open reading frames, so they're really using a huge amount of the genetic material in each of these individual ones. So this is how this is. I'm not going to ask you all the details on these. I can't remember how you make the particular L1 messenger RNAs, and I can't remember which one of these is coding for which protein, but that we have poly-A tailing that happens at 5 different places, and then differential splicing that happens in each of those, giving you different proteins. And again, all of these are late proteins. All these are the things which are going to end up in the virion or involved in that process. Yeah?
The poly-A-tailing, is that within the genome? Is it encoded within the genome?
No. So poly-A-tailing is regular cellular poly-A-tailing. So the CPSF, CSTF, regular binding to and association with these particular transcriptional termination sites too, because as soon as you put on poly-A, you're going to be terminating transcription. These were how they were originally found in the first place, was studying the major late genes in adenovirus, was all the binding sites for all of these things. Okay, how do you get everything together? Basically, it's a lysis process, and we have a wonderfully named protein here. It's the early 3' transcript, but it's made as a late protein. It's the death protein, kills off the cells, and also this transcript in E4, and sorry I didn't emphasize this here, but the E3 genes are here, E4 is right over here. E4, these are, again, still early genes being made on the opposite strand from the E4 promoter. This is a great protein in terms of killing off cancer cells. The open reading frame 4 seems to be very specific for killing cancer cells. We don't know why that is, and people have been trying to exploit that in terms of cancer therapy. The other process, again, as I mentioned, apoptosis. Apoptosis is going to be blocked by what? E1B, and that E1B protein in particular is going to be important for looking at some of these cancer and gene therapy processes. So we've assembled, we've gotten outside of the cell. That's it for regular adenoviruses. Let's talk about how to tweak adenoviruses in terms of some fun stuff, including gene therapy processes. So a couple of different things. Let's start here down with tumor therapy. No E1B. If you don't have E1B, what happens? Apoptosis, because E1A is going to stimulate that apoptotic process. If you have P53 around, you're going to have normal apoptosis. Those cells are going to die. If you don't have P53 around, which is true in many, many, many tumor cells, then, in theory, this virus can now replicate perfectly happily because you're not going to undergo apoptosis. And one of the things that cancer cells don't do is undergo apoptosis. But then you'll be expressing your E4 protein and killing off the cancer cells. Great, wonderful idea. Was a, probably got all the way through phase 3 clinical trials. Did actually have some effect. In fact, people are now still using this in China as sort of secondary or with chemotherapy treatments in order to get, and it does seem to work reasonably well. The problem is we don't quite happen to know. It turns out that these viruses that are missing E1B actually work quite well in P53 positive cells. So why aren't they causing apoptosis in those cells? It's still a very open question. So do you want to have a drug that works even though you don't know how it works? Is that something you want to be using? The FDA says no. The Chinese say yes. There's no good answer to that question. I mean, there's really no good answer to that. On the other hand, people are using many more adenoviruses now in the US, also in clinical trials, where they're completely missing the E1A and E2A proteins. And most important here is you're missing E1A. If you're missing E1A, you're not stimulating the cell cycle at all. And so these are going to be adenoviruses that can get whatever you put inside them to the nucleus and have them in there, but the expression of the cell cycle is now going to be very different. So you're basically getting that one particular gene in there. And that's partly because these adenoviruses do bind to receptors in lots and lots of different cell types. And so you can get that going in, and getting that actual targeting is still a big issue and a question on how to work with that. The fat virus, I promised to talk about the fat virus. Adenovirus 36, about 15 years ago, was shown 16 years ago now, that it increases adipose tissue. adenovirus 36 antibodies in 30% of obese people and 11% of non-obese people. People got really excited about this. Here's a notice where it's from, whether we believe this source or not. Interestingly enough, in this particular news article, which was, I think, in 2007, mentions where these people are. And you go to their website, they actually don't list that person as being there anymore. And strangely enough, all of the papers published on this have one author in common. So I think it's rather open whether there's any particular connection with that. Yeah?
This is just shown to increase adipose tissue, not like form lipomas or, basically what would be considered a tumor for fat. Do you know?
If you like, you can take a look at some of these papers. But I think the connection is tenuous at best,
Okay.
I would say. And one of the latest ones here is looking at, this is the latest paper I could find in 2010. 78% were adenovirus 36 positive and obese, 7% were positive but not obese. But look at these numbers, 15 and 4. The numbers here are incredibly small. If you want to hear a little bit more skepticism about this, listen to TWIV 18 and TWIV 101, where they talk a lot more about this. So I don't think that there's good evidence for it may or may not be involved. And as I mentioned before, we've got hundreds of adenoviruses out there that 15 versus 4 have these, adenovirus positives or not. I think is a very open question in terms of a connection to disease. But it's one of the issues that we have when you particularly got these viruses that are everywhere and diseases that are everywhere, how often they can be crossing with each other. Yeah, Trevor.
How do they exit the cell? Do they lyse?
Yeah, so the death protein actually does cause lysis of the cell.
How is that useful when you're trying to treat a person? And you give them gene therapy and then it's...
Right, because those genes don't get turned on. Remember, E1A is not only a protein which is stimulating the cell cycle, it's also a transcriptional activator. And so you need to have that transcriptional activator in order to turn all these genes which are going to lead to cell lysis. So it's basically all that you're doing with these gene therapy vectors is getting that gene in. They're not trying to get massive amounts of replication or anything else going on there. So it's not a process of trying to get it to spread. Just wanted to quickly finish up here with adenoviruses that do seem to cause diseases that people care about. Adenovirus 4 and adenovirus 7, strangely enough, in boot camp, seem to have huge numbers of recruits coming down with adenovirus diseases. This was causing a big issue for the U.S. military. And so what they did was immunize with an oral route. Now, adenoviruses are usually going in through aerial routes, but here, apparently, if you just give powdered virus in the gut, you get over 100% reduction in these viral diseases in these military recruits. And so if you go and look at the CDC website looking for adenovirus vaccine, it's approved for active members of the military from 18 to 50. It's the only group of people that it's actually approved for. But it seems to work incredibly well. They discontinued it. They saw these big spikes in adenoviral disease, and then brought in this vaccine, went way down. But that seems to be the only case where you're getting this kind of real disease formation by adenoviruses, which you can prevent with these vaccines. And it's really interesting that literally all you have to do is put them in a pill and in the gut, and you get antibodies which will protect you from. So we'll stop there. We won't do our second clicker question. Only one clicker question today. Oh no. Probably no clicker questions on Wednesday. We'll have a ton on Friday, don't worry.